Okay, so yes, I'm Andrew. I'm from physics in the School of Science, and I guess I'm here because I used Figshare earlier this year, about when the institutional form of Figshare came online for RMIT. Sam gave me a lot of help with that, and I guess I'm just going to tell you a little bit about how data is used in the research community that I work in, and in particular, there's really a lot of data in this community, so it's just a bit of a case study, really. Okay, you want to go to the next slide, Sam?
There we go.
So I guess first I'll just give you a little bit of background about the scientific field that I come from. And essentially it's a field that uses very large X-ray facilities like the Australian Synchrotron, which is down in Clayton in Melbourne, just over the road from Monash. Or I think the other ones that I use is some international facilities called X-ray free electron lasers. These facilities are really large. The Australian Synchrotron is about the size of a football field. And the one that you've got down the bottom is a thing called the Linnet Coherent Light Source, which is in Stanford in California. It's about 3 kilometers long. And without really going into the technical details, basically what these machines do is they have big accelerators and they speed up electrons to very, very high speeds, put them through some magnets and make very bright X-ray beams. So they're basically very large X-ray light bulbs. That's kind of what they are. Do you want to go to the next slide, Sam?
Yeah.
And what we use them for is this, I mean, there's heaps of uses really spanning, material science and biology and going up to medical imaging and all sorts of things. But a lot of, I guess, what I use them for is really to get down to the atomic details of materials and proteins and things like that. And some of the data that we actually measure at these facilities, particularly the type of experiments I'm involved in, it involves taking a camera, an X-ray camera, and taking a kind of picture of where the X-rays go after they have interacted with the material. And the example you've got there on the right is an X-ray pattern that we collected from a protein nanocrystal. As you can see, the actual pictures we actually collect on the X-ray detector don't really look like the thing we took the picture of, but that's kind of what we analyze them and we figure out either an image or we determine an atomic structure or something like that, and that's where a lot of the physics actually comes in. But I guess the point of showing you this was just to show you that the kind of data we're talking about are images, like they're actual images taken on a particular camera. Another aspect of these experiments is that access to these facilities is usually free. Most of these facilities are government built, and there's many different countries around the world. They all pretty much operate on the same principle, which is that the government builds them for, usually sometimes with some industry partnership, but they build them mostly for fundamental science. So usually the researchers might have to pay for, travel costs to get to the facility. They might have to pay for any materials they want to bring to the facility, but otherwise it's actually free access. The flip side, though, is it's competitive. So we have to put in proposals to do experiments, and they get ranked and they get peer reviewed, and then the best proposals get the access to the facility. Can we go to the next slide, please? So the X-ray free electron laser facilities are probably where we get the most data, and it's because they produce basically hundreds or thousands of these images per second. So if you kind of imagine running your camera at sort of 1,000 frames a second, that's kind of what we're doing. And your experiment might go over several days. So you might have kind of 50 hours worth of experiment, and you can very easily rack up tens or hundreds of terabytes of data in one of these experiments. Not all of it is useful, but all of it is collected and all of it is stored, and all of it has to be kind of analyzed by the researchers to figure out, which is actually the best data to determine or to test the scientific hypotheses that they were setting out to collect. So this particular community that uses these particular international X-ray facilities has a kind of a data. I mean, data is actually very important, and data management, I guess, became very integral to how the actually community runs and how the scientific field runs. Because these facilities are actually free to access, but the amount of data you can generate is so large, it's not actually realistic to expect every user group that comes for just a few days to have the resources back at their home institution to actually take all the data back home, to store it, to manage it, and all that sort of thing. So actually, it's built into these facilities that they would have basically servers on site, and they have data storage on site. And you usually access a lot of your data, at least the raw data, you would access remotely by effectively remotely logging into a server. Yeah, so that's kind of mostly how it works. And then, yeah, what you might do is that after, I guess, you've analyzed some of your data, you might find a subset of that data which is more useful, and then you might take that back home, and you might analyze it further and turn it into figures in a paper, and then eventually publish a small part of your data. Can I go to the next slide, please, Sam? Yeah, so I guess in this community, this is kind of how, I mean, it's not the data management life cycle, but it's the sort of data life cycle in the scientific kind of thing, is that you would start out trying to get access to a facility, and you would write a proposal. That might take a few months to get reviewed, and if it is hopefully accepted, you might have to wait another few months before you're actually scheduled at the facility. So, the better part of 6 to 10 months has sort of already passed before you actually get to start your experiment, and you might use some of that to actually prepare what you need to do the experiment and access your materials and buy your materials and things like that. Then your experiment might only go for sort of 2 to 5 days, but in that time you collect your kind of tens of terabytes of data. Then, because you've really got a lot of data, it's pretty standard for the actual data analysis to take up to a couple of years before you've really sure that you've got your kind of scientific result, at which point you can publish your paper and you publish your data very often in this field. And then you've actually got your material for your next proposal. And so, then you would go and put in another proposal and you would do the whole thing all over again. Can we go to the next slide, please? Yeah, so the community actually set itself up with a database to store the data for itself. So for a particular class of experiment, for a particular type of imaging experiment, particularly for proteins, this was set up in Berkeley in California. I think it's the Berkeley National Lab over there is where it's housed. It's the current X-ray imaging database. And it was really pretty good that this thing was set up early on, because access to some of these facilities, particularly the international X-ray laser ones, is so limited. The only way that really the community could grow was by sharing data pretty much from the start. You're collecting these very large data sets, and for other groups who didn't have the fortune to be there at that first experiment or to get time on the experiment, sharing data has actually been really excellent way for us to grow the community. It's very easy, if you've got a new PhD student or a new postdoc and they're trying to learn the techniques, learn how to analyze data, so that they can go and get some real source data and some raw data and work with it to learn. So CXIDB was pretty much fulfilled that purpose for this community. They standardized the data format using a HDF5 format. I guess for anybody who might not be familiar with that, it's actually a very flexible format where inside the file itself, you can basically organize your data and all your metadata a little bit like folders on your computer. So you set up data sets inside the file, and you set up metadata kind of labels, and you can basically organize your data inside your file. And so this particular database, they set up a standard data format, which for everybody to use when they upload. Then they gave the data set a DOI, and they link back to the papers where it was originally published and all that sort of stuff. And so this has actually been really valuable for us. Yeah, and I think, yeah, we'll go to the next slide. And I guess the other one is, I guess the last thing I'll just mention is that for smaller data, so I guess the CXIDB was really good when the data sets get up to kind of a terabyte. But I've also got other science where I work on just sort of data sets that are more like gigabytes. And for that, for a couple of publications this year, I was able to use Figshare at the RMIT Figshare account. And I would just say that I actually really enjoyed using this. I thought it was really easy. It was a really easy interface to upload the data. It was nice that one could set up the data set with the DOI before you publish it and delay publication, so that you could time the publication of your data to come out with your article. I mean, obviously, I guess it was mentioned earlier on that journals are not so worried about that you can publish your data earlier, but it was still nice for me to be able to do that so that I could finalize the data set and the manuscript at the same time. Yeah, it was a really great thing to use. So I'm actually really glad that RMIT has done this and provided Figshare for us to use.